Hello students, welcome to the fifth lecture of the chapter Molecular Basis of Inheritance. I'm Mouchandra Paul, so let's begin. So in the previous class, we talked about the process transcription, all right? So we saw that in transcription, what is transcription basically? The process of copying genetic information from a certain segment of the DNA into RNA. Here we saw why only one of the DNA strands gets copied, not both the strands, right? With an example. Then we looked into what all components are there within this nucleus that does the transcription process. Like there is polymerase enzyme, RNA polymerase, and the mRNA that will be formed, right? We need a DNA template, etc. And also we looked into what is the structure of transcription unit. So the region that is going to get transcripted, in that region we saw at the beginning at the 5' end there is a promoter region, right? And 3' end there is a terminator region, and in between it is the sequence of the gene that will get copied, right? Then we looked into what all types of RNAs get formed during this transcription process. So there are mRNA, tRNA, right? Then rRNA. So there are three types of RNAs. And lastly, we looked into in detail the transcription process. The process consists of three steps: initiation, elongation, and termination. All right. So one by one, the RNA polymerase reads the bases that are there in the DNA and adds the base that is complementary to that particular base, and then moves forward. So in this way, the transcription process happens, and after it reaches the terminated zone, it the RNA polymerase gets removed. So for transcription process, there are certain factors, right? We looked into what are the roles of that factors, like sigma factor, rho factor. So this is a short recap on what we studied in the previous class. So in this class, we will look into the process of translation. That is the process of synthesizing protein or polypeptide chain from mRNA. So before understanding translation, we should first understand what do you mean by genetic code or codons. What are codons? Okay, so let me begin by raising a question that why genetic code is actually needed. Why do we need genetic code? All right, what role does it play? So we saw in replication, right, that nucleic acid, that is DNA, information in the DNA gets copied into another DNA molecule, correct? So the language is same, all right? The ATGC, it is a basis, it is present in the nucleic acid, and here after it gets replicated, the DNA molecule also have ATGC. So here we can simply use the complementary base pairing rule, right? And get a copied DNA molecule. Same with translation as well. Here also a segment of nucleic acid, DNA, right? Whose language is again in form of ATGC. It gets uh, transcripted, okay, to form an RNA molecule. Here only difference is there is U instead of T. So in RNA there is AUGC. So the nucleus or the machinery for transcription, it has evolved and it understands that it needs to add U in place of T. So the machinery, the evolution has actually developed this machinery quite well. Okay, in a help that helps in recognizing that here now U needs to be added instead of T in RNA molecule. But in protein, uh, it becomes complicated. All right. Okay, so this is the thing, right? So in transcription and in replication, the language is same. Okay. Now next is in case of translation. In case of translation, here what happens is the language becomes different. The nucleic acid, that is mRNA, all right? The whatever information is there in mRNA, that gets copied, okay? And in form of amino acid, okay? So this language becomes different. Here, mRNA understands sorry, not AUGC, and in proteins, there are around 20 amino acids. So here is why, genetic codon or genetic codes plays a very important role, okay, in converting or translating uh, what a segment of mRNA means in terms of amino acids. That is why here cell needs the genetic codon in translating the two languages from one medium to another medium. Now, people found it very difficult, to understand the basis of the genetic code. Okay, to understand or to prove, does it really something called genetic codon that exists in cell? So people from different disciplines like physicists, organic chemistry, biochemists, geneticists, they all had to come together and work together to understand the basis of the genetic code. So there was a physicist named George Gamow, okay, who proposed that who proposed the following. Okay, so he said in nucleic acid, that is mRNA, there are four bases, right? We know these are A, U, G, C. These are the four bases that are there in nucleic acid or mRNA. Whereas protein, a polypeptide chain can be composed of 20 amino acids or a combination of 20 amino acids that they knew at that time. So what he proposed was a genetic code could be something that is composed of a combination of bases. So few bases together, okay, say AUGC. So these four bases together can uh, form one genetic code. This is he said. Now he also said that the code should be made up of three nucleotides. Okay, uh, at that time it was a very bold statement that he said only three. Okay, not four or five or six. He said that the only three nucleotides together, any three nucleotides means any three bases together would form one combination, and this one combination is a genetic code that is going to signify one particular amino acid. So this was a very bold proposition compared to the knowledge that was there at the time of the discovery. So according to George, if we apply his proposition that a combination of three nucleotides or three bases forms genetic code. So using permutation and combination, we can actually determine how many such codons can be there. All right. So he said there can be a combination of three bases. All right. So we can then say that there are three positions. Fine. And at each position there could be any of the four bases, right? Here also any of the four bases can come here. Here also any of the four bases. So if we do this multiplication, and then we'll see that in theory there are 64 codons or 64 such combination of nucleotides or bases is possible. Okay, that actually specify each of the 20 amino acids. But here, the numbers are not matching, right? There are 20 amino acids and there are 64 codons. So number of codons is greater than number of amino acids. It is only 20 and here is only 64. So then people obviously can imagine that many people opposed to this idea, right? But later on, his proposition was proven true. Okay. So how it was proved? We will not go into the details, but we will see who all contributed to this, proving this particular proposition. So Khorana, he was an Indian scientist. He developed a chemical method for synthesizing RNA with a defined combination. All right. Uh, and also people used his method for synthesizing artificially uh, mRNA, okay, in lab. Okay, they can make this mRNA of any sequence they like, okay, and use that as a template for translation. Then Nirenberg's work uh, helped the people to use his method of synthesizing proteins outside cell. So he described a particular method which people can use to synthesize synthesize proteins, but outside the cell, like in artificial in vitro environment outside the cell. So people used his method to synthesize the proteins of any sequence. So uh, Khorana gave the method for synthesizing RNA. Nirenberg gave the method for synthesizing protein. And there are also enzymes, okay, called polynucleotide phosphorylase enzyme that helps in synthesizing RNA, the defined sequence, in a template independent manner. So in transcription, we need a template, right, from which the information get copied. But this particular enzyme actually helps in synthesizing RNA molecule without a need of a template. And this RNA molecule would have a sequence of our desire. Whatever sequence you want, you can put in there. Okay. So this was a enzyme mediated synthesis of RNA, whereas Khurana proposed rather chemical based. So these three methods that were proposed by different scientists, when these methods were put together, um, people could prove that yes, indeed, genetic code is there inside the cell, and it actually is in between, is in between, like act as a mediator for copying of information from mRNA to protein. So one can think of like this is an mRNA, okay, and it is composed of genetic codes. We can break down the sequence of mRNA into genetic codes, and these genetic codes can be read by particular machinery to form proteins or polypeptide chain. So this is the translation. So this genetic codes are a language, you can say a new language of a new language of understanding genetic information. Okay, you can think of genetic codes like that. It is helping in translating the information that is there in mRNA into a sequence of amino acids that is going to be forming a polypeptide chain. So that is the significance of genetic codes, and now you must have understood why genetic code is needed. Okay. Now here is a table that I've taken from the NCERT book, which is showing the combination of all different genetic codons that are there. Okay. So one can read this table like this. So this this first column is showing the first position of the so we know a genetic code comprises of 3 nucleotides or 3 bases. This is the first position of that particular codon, and this this next four columns, 1, 2, 3, 4, 1, 2, 3, 4. These four columns is suggesting the second position of this particular codon. But the last position is shown by this last column. So in this each of these column and then rows, you can see bases are written, right? So since we are going to translate the information from mRNA to protein, so the bases that are there in mRNA are A, U, G, C. So exactly that I have read. So I have written here. So this is A, U, G, C. So here also A, U, G, C, here also A, U, G, C. So on all three sides, we have written A, U, G, C. So how we can read it? This U, and then this U forms U, U, U. And this particular codon codes for the amino acid phenylalanine. Okay. Similarly, if you see here, this particular codon, C, A, U. So it has made a codon CAU, which codes for the amino acid histidine. Similarly, here also C, look at this codon, C, A, and then U, CAA, GLN, okay, that codes for, I guess, glutamine. So these are the, where LEU, PHE, these are the three-letter codes for the amino acids. Similarly, you can read here for the alanine, look at this particular code, G, so here C is there, GC and U, GCU. So in this way, you can easily understand the table and find all the possible 64 codons for various kinds of amino acids. Now, you should also remember this table as well, which is showing the names of all 20 amino acids. Okay, this is the name of all 20 amino acids. And each of the amino acid can be written in form of three-letter code. Like for glutamine, the three-letter code is GLN. And also we can assign a one-letter code to them. Okay, so most of the time you will see that the first letter of the name alanine would be the A. So A is the one-letter code. But since there are also alanine, arginine, aspartic acid, so these are for all four amino acids starts with A. So it is not possible to assign one-letter code A to all of them. So people similarly assigned different letters of the English alphabet and made a one-letter code. So these codes. The three-letter code, one-letter code for each of the amino acid is universal and used everywhere in every kind of literature and everything. So you will have more use when you will go into higher studies. But for now as well, when you are going to solve few questions, you should remember the codes, three-letter codes or one-letter code of few common amino acids. You should know them. At least from the three-letter code, you can understand what will be the name of the amino acid, like MET. Okay, so this is going to code for the amino acid methionine. But you don't need to remember all these 64 codons. You don't need to do that. Only you will need to remember 4 important codons. What are those? I will describe a little later. So these are the 64 genetic codons, okay? And each of the 64 genetic codons codes for various kind of amino acids. This is the table showing the names of the 20 amino acids and their respective three-letter and one-letter codes. So what are the features of this genetic code? Okay, first thing, it is a triplet. Means it is a combination of 3 bases. Like we saw U, right? Then A, U, G, etc. A codon cannot be more than 3 or can be less than 3. It has to be a combination of 3 bases. Then there are out of 64 codons, there are 61 codons which codes for amino acids. Rest 3 codons, they codes for stop codons. So what are stop codons? So here in the this table, look at the codons that I have made a box around. So U A A, U A G, U G A. These are called stop codons. Okay. So whenever these stop codons are there in the mRNA, at that point the protein synthesis will stop. Okay. Hence the name stop codon. So UAA, UAG, UGA. So these are the 3 stop codons. UAA, UA, UGA, and UAG. Now, each of these stop codon doesn't code for any amino acids. All right. Then one amino acid, it can be coded by more than one codon. Okay. And this particular property of codons is called degeneracy, or it is often said that codons are degenerate in nature. Remember the term degeneracy or degenerate. Okay. So degeneracy or degenerate means that one amino acid can be coded by more than one codon. So here in this table, you will see that phenylalanine, PHE, is coded by 2 codons, right? Similarly, C for serine, it is coded by 4 codons. Then proline, it is coded by again 4 codons. So each of these codons, CCU, CCC, CCA, CCG, all these 4 codons codes for only one amino acid, it is proline. So this particular property of genetic code is called degeneracy. Next, when the machinery, translation machinery reads the sequence in the mRNA, it reads in a contiguous fashion, or it contiguous means there is no punctuation in between, there is no full stop or comma or anything like that. The reading happens continuously. Okay. Next, the codons that we talked about, 64 codons, these are universal. Universal means these codons are same in human, in bacteria, in virus, or in every living organism, okay, that are known to mankind. So that is why one can find out the protein sequence that is present in human or present in rat or present in a fish, okay, because the language is same, genetic code is same, it is understood, and when once you understand this particular genetic code language, you can understand the sequence of the protein or the sequence of the amino acid in every living organism, okay, in the planet. But there are few exceptions that people have found. One is the genetic in mitochondria, the genome is a bit different, okay. Mitochondria has its own DNA and synthesizes its own RNA and synthesizes its own protein. So in mitochondria, people found that few of the codons are different. Also, in few kinds of protozoa, there are also few 1 or 2 genetic codons, they are different. So these are 2 exceptions. Other than that, in all living organisms, the genetic codon, 64 genetic codon is exactly the same. Okay. And that is why we can understand through genetic codon, the sequence of protein, sequence of mRNA, etc. Now, another feature is that the AUG, this is the codon, AUG, it has a dual role. One, it codes for the amino acid methionine, and second, it is also called as the initiator codon. Initiator codon means wherever this in the mRNA sequence, wherever this AUG sequence will be there. So if this is a mRNA strand, okay, and say AUG is here, okay, the AUG is here, and in before a in the upstream in the towards the 5' direction, there is no other AUG molecule. So the protein synthesis will start here where the AUG is located, and it will continue in this direction towards 3' direction. All right. So whatever sequence was there upstream of AUG, that doesn't get translated. So that is the role of being initiator codon. Wherever initiator codon is there, from there onwards, the synthesis of protein will begin. Okay. Now, let me give you a short exercise to do. I request you to do it along with me. So here, this is a mRNA sequence that is given. This exercise is given in the book as well. So here's a sequence of the mRNA. So from this mRNA sequence, you have to predict what will be the amino acid sequence or the sequence of the polypeptide chain. So you have to use the knowledge of genetic codes or codons that you have learned so far. So here, AUG. First, you have to break down the entire sequence in combination of 3 bases at a time. This is one base, one combination, UUU. This is another combination. So I am basically starting from here, from A onwards, because wherever AUG is there, from there onwards, synthesis of protein will begin, or the addition of amino acids will begin wherever the AUG sequence is there or the initiator codon is there. So we have to also read in from AUG only. We cannot read from this end. Okay, we have to start reading the sequence for making the amino acid sequence from the AUG end. So I'm just breaking down the entire sequence in form of 3 uh, nucleotides at a time. In this way, here also I'm breaking like this. So suppose I have done it. Now you have to look at the table, this table. You have to look at this particular table, okay, and see which of these codon codes for which kind of amino acid. So here AUG, we know it codes for the amino acid protein, methionine. Triple U or UUU, it codes for phenylalanine. The phenylalanine is also coded by another codon, that is UUC, right? So here this is phenylalanine, UUC is again phenylalanine, PHE, UUC phenylalanine. Similarly, in similar way, we can do for the entire mRNA sequence, and we'll find that this is your sequence of amino acid that is asked in this particular question. Okay, try to do it yourself. It is quite easy, and you have to use a genetic code table in order to come to this particular answer. Now, if we do the opposite, here what we did here from the mRNA sequence, we predicted the amino acid sequence, right? What we go in the opposite direction from amino acid sequence, it is given to you, and from amino acid sequence we have to predict what will be the mRNA sequence. So suppose this is your amino acid sequence that is given. Now let's look at the again we have to use the genetic codon table in order to get the genetic codons for each of these particular amino acids. Now MET or methionine is the start is the amino acid that is coded by the start codon AUG. Then there is PHE. Now PHE can be either coded by UUU or UUC. All right. Now here also PHE. So here also UUU or UUC. So you get my point, right? So in this way, we can predict what will be the mRNA sequence. So these are the possible mRNA sequence. In one combination that there can be all triple U's. All the phenylalanine molecules can be coded by the triple U. Another way can be that all the phenylalanine molecule can be coded by the UUC, okay, UUC, or it can be like random. Few phenylalanine are coded by triple U, few are coded by UUC, so it can be random. So these are the possible types of mRNA sequence that is we can predict from the given protein sequence. So what you understand from this particular example? First thing is we are using the degeneracy property. Degeneracy property. What is degeneracy? It means one amino acid can be coded by more than one codons. So here phenylalanine gets actually is represented by both UUU and UUC. So we are using the degeneracy property. Another thing to note is one codon for one amino acid. Okay, so triple U, it can only code for phenylalanine. It cannot code for any other amino acid like serine or alanine, etc. So these two things we should keep in mind while doing such kind of exercise where we are uh, predicting the sequence of one when the another sequence is given, right? So these two important things you should remember, that codons are degenerate, means one amino acid can be coded by more than one codon. Another thing is one codon can only code for one amino acid. Okay. Now, now once we have understood the genetic codon, okay, what genetic codons are, why it is needed, okay, how it is actually helping in translating the sequence in mRNA into a amino acid sequence. So now the understanding of mutations that we saw in the previous chapter, right? We know mutation is a permanent change in the base sequence of a DNA, right? So we mentioned two types of mutation in the previous class, in the previous chapter actually. First is the point mutation, where there is change in the single base pair. That is one base pair gets substituted by the another, by another kind of base pair. And another was the frame shift mutation. So keeping that definition in mind, we can actually apply that, okay, and see that how one particular change in uh, base can make the can change the meaning of the entire sequence. So suppose this is a statement, okay, and it represents a DNA sequence, suppose. Okay. Now instead of writing this in form of AUGC or ATGC, it is written as like Ram has red cap. Okay. Now here, because of some mutational event, this particular base, say R, okay, gets changed. So if it gets changed, the meaning of the sentence is changed to T. So now the meaning of this sentence get changed completely. Now it may be possible that this particular statement doesn't make sense. Same with the sequence of the DNA as well. So if there is a particular change in the base pair, the now the meaning of that particular DNA sequence gets changed completely. And when that particular sequence, the DNA gets transcripted and then translated to form a protein, the structure of the protein, or whether or not the protein is functional or not, okay, that gets, becomes uncertain. In some cases, even if there is a change in DNA sequence, the resultant protein structure, it can be functional, okay, by random. It is a completely random event. It happens randomly. You cannot, or the cell cannot control it in any way. This mutation can happen anywhere and at any point of time. So in sickle cell anemia, where it is a disease caused because of point mutation, okay, so there a single base pair change happens, because of which a particular amino acid completely gets changed. So because of that, the structure of the protein gets changed. Now the new structure of the protein is not at all functional. Because of that, a disease like anemia happens. So that is the repercussion of a simple event, that is point mutation, and how it can affect the structure of the protein or the sequence of the amino acid. Now, uh, what is frame shift mutation? So here what happens, due to insertion or deletion of one or two or multiple bases, the way the machinery, transcription machinery or translation machinery reads the sequence, that gets changed. So we can again understand that using a particular sentence. So say this is your sentence, Ram has red cap. Now if you insert, say, a letter F, okay, where here say I'm inserting a letter F here. Now how the sentence gets changed, let's see. Okay, the meaning of the sentence get changed. Okay, uh, because of insertion of the letter F at any random point in the sentence. So when we divide the sentence into three letters at a time, the meaning gets completely changed or doesn't make any sense. Similarly, we can insert another letter, say, here. We can insert, say, G. Now the sentence become something like this, right? The meaning of the sentence gets changed. When we divide into three letters at a time, it doesn't make any sense. It doesn't make any sense to the translation machinery. Now again insert another letter at any random point, say here, all right. um S, suppose let's insert a letter S. So here what happens, this. Now if you notice what has happened, b- at the point of insertion of the first letter F, subsequent sentence, the meaning of the subsequent sentence gets completely altered. All right. Here, now when we insert another letter, say G, so from that point onwards, the meaning of the sentence gets changed. So here I have inserted G here. So here the meaning of sentence gets changed. Then we have inserted another letter, say S. So from this position onwards, the meaning of the sentence gets changed. So so the frame shift is happening. Okay, this is your frame. This frame gets shifted wherever there is an insertion of a new particular base. Okay, so that is called frame shift mutation. That frame shift mutation is happening because of insertion. So at the end of inserting three letters, what we see is that a new word gets added. A new three-letter words get added in between. Now, suppose it were an mRNA sequence. So in that case, it would have been an addition of a new amino acid within the polypeptide chain. And that particular polypeptide change, how it is going to function because of the addition of this new amino acid, that no one can predict. Okay, it can even be like non-functional. It doesn't do its job, or it can give rise to a new function. So there can be a loss of function or a gain of function. So if the gain of function is towards the positive direction, like it is helping in survival, so the organism or the person who is having that particular mutation, it will get passed on to the next generation. Now if the addition is such a way that it is harming that particular individual, it is causing a particular disease that is deadly, so it will not get passed. So that is a basis of genetics, and how genetics and this concept that we are studying is related to each other. Now in another case, the frame shift mutation can happen when there are deletions. So let's consider the same statement. Ram has red cap. Now let's delete one uh, one letter at a time. So let me delete the letter R. So here how the frame is shifting. Let's see. Here R is deleted. So E will shift in this direction. EDCAP. Okay. R is deleted. So the meaning of the sentence gets completely altered. Now say E is deleted. Now D will shift one place backward, right, in this direction. DCA, then P. Now suppose D gets deleted in the next step. So what will be the meaning of the sentence? It will be cap. So here what it means is after four mutational events, actually three mutational events, the meaning of the sentence get completely altered. Here the sentence becomes Ram has cap. So here it is happening because there is a shift in the frame. That is how the translation machinery is going to read the particular sequence of the mRNA that gets shifted because of the deletion event. And when this particular sequence will get translated to form a protein, now that protein molecule will not have one particular amino acid, one whole amino acid. Now it can then again negatively or positively alter its function. So that is about point mutation and frame shift mutation. And we understood it through the, example of how one particular base pair or multiple base pair gets added or deleted through a simple English statement. Now, let me introduce you the new role of the tRNA molecule. That role is of an adapter molecule. So what is an adapter molecule basically, and how it came into the picture and why it is important now? So the Francis Crick guy, he was one of the guys who discovered the DNA molecule, right? There was Watson and Crick, the famous Watson and Crick who discovered the DNA. So um, Crick, he proposed that there must be a particular molecule, okay? He termed it as an adapter molecule that would do two functions, okay? Or the structure of the molecule is such a way, would be such a way that it would do two functions. First, it can read the code, genetic code that is there in the mRNA, and on the other hand, it will bind to a particular amino acid. Okay, so when Crick proposed this particular presence of a particular adapter molecule, at that time tRNA molecule was known to people. So people knew that there are three types of RNA molecules: mRNA, RNA, and tRNA. But tRNA was called at that time as sRNA or soluble RNA. Okay, transfer RNA, the name transfer RNA came later on. So people later on discovered that tRNA is actually this particular adapter molecule. Hence, they changed their name. So why the tRNA, or how the tRNA is able to do these two things? Let's see a structure first. So this is a particular mRNA sequence running from 5' end to 3' end. Okay. So there are genetic codons on this particular mRNA sequence like AGU, UAG. These are the particular codons. And on one end of this tRNA molecule, this is a tRNA molecule, the cartoon depiction of that particular tRNA molecule. So at the one end of the tRNA molecule, there is a combination of three bases. Okay, and these three bases, they are complementary to the uh, bases that are there in the mRNA. So this is called a codon, and the sequence that is complementary to this particular codon is called anticodon, right? So you can see AGU is one codon. What will be complementary to this particular sequence? It will be UCA. This is anticodon. So at the one end of the tRNA molecule, anticodon is there, and this is the region that binds, region that has that anticodon is called anticodon loop. Whereas at the other end of the tRNA molecule, there is an amino acid that is bound. So this end is called amino acid acceptor end. So AGU, this is a codon, right? So let's check in the table what this codon codes for. So it AGU, AGU. This is AGU. Now AGU codes for serine. Okay, it codes for the amino acid serine. So we see that this tRNA has brought the amino acid serine. All right, and and this particular amino acid has to be present over this particular codon. In order to do that, the anticodon is there, which is initiating this temporary hydrogen bond between the codon and the anticodon. Similarly, there is another tRNA molecule that is bringing the amino acid tyrosine because it has read the codon UAC. You can check whether or not UAC codes for tyrosine in a similar way that I've showed you just now. So that is the role of the tRNA. One end of the tRNA has the anticodon because of which it can read the codon on the mRNA. The other end is bound to the amino acid, and this amino acid is actually corresponds to that particular codon. So that is why the name transfer. It helps in transferring the information from mRNA to protein. Now the structure of the tRNA is like of a clover leaf. So in the previous image, the structure was not very accurate. So this is a accurate description of the structure of the tRNA molecule. So the structure is like a loop. So this end is called anti-loop codon, all right? Anticodon loop. This end is called anticodon loop. So this is your GAA. This is the anticodon for the codon CUQ, correct? This is the anticodon. This will be codon. Then just opposite, vertically opposite is the acceptor, amino acid acceptor end. So here an amino acid can bind that corresponds to the codon CUU. Now for each amino acid, there will be a specific tRNA molecule. So there will be a tRNA molecule for serine, there will be tRNA molecule for tyrosine, and each of these are unique. They cannot, a serine tRNA molecule cannot bring the amino acid tyrosine, okay? It can only bring the amino acid serine because it has the anticodon to bind to the codon for the serine. Similarly, uh, true for all the amino acids. Now, initiation, that is start of the translation, is done when the initiated tRNA comes and binds to the first codon or the start codon or the initiated codon on the mRNA. You know what is initiated codon? It is AUG. So for AUG, there will be an initiated tRNA with the anticodon UAC, which is the anticodon, okay? And this particular tRNA will bring the amino acid methionine. So from there onwards, the synthesis of the amino acid sequence, that is synthesis of the polypeptide chain, will begin in the forward direction, that is towards the right side, all right, a 3' end, towards the 3' end. Um, now when termination is going to happen, like now the sequence of the amino acid has reached towards the end, okay? So how that termination will happen? It will happen not through tRNA. Okay, it will not happen through tRNA because at the end you will find a stop codon, right? Like UAA, UGA, or UAG. Now, for this three types of stop codon, there are no tRNAs. It happens through binding of another kind of a protein or another kind of a molecule. So for termination, there are no stop codons. There are no tRNA molecules for the stop codon. And for initiation, there is an initiator tRNA. Now let's describe the translation process. So we know what are genetic codes. Okay, we know what is the structure of the tRNA, or what is the role and what is the role of the tRNA molecule. We are aware of these two things. So now we can easily understand how the translation happens. So translation is basically a process of polymerization of amino acids to form polypeptide chain. All right. Polypeptide chain is formed because information in the mRNA gets translated. Now the sequence of the amino acid in the polypeptide chain would follow the sequence that is there in the mRNA. So we saw in the mutation example, we start reading, right? We start reading from the 5' end where there is AUG, and we move towards the 3' end, and we go one combination, that is one code at a time. So if there is first a code for AUG, the first amino acid will be methionine. If the code for alanine is there in the mRNA, the next amino acid will be alanine. So similarly, this synthesis of the amino acid sequence, the synthesis of the polypeptide chain will happen. Now, for formation of the polypeptide chain, you want peptide bonds to be formed between two adjacent amino acids. Now, these peptide bonds needs energy. Okay, it requires energy to be supplied that will increase the activation energy of the reaction, and this does allow the formation of the um, polypeptide chain or the peptide bond between two adjacent amino acids. And so for that, what happens is it is quite unique. Here we don't need ATP like molecules during the process. Here what happens that the tRNA molecules, they get charged. What do you mean by charge? What do I mean by charged is that when a tRNA molecule gets to the tRNA molecule, amino acid gets attached. Amino acid gets attached towards the amino acid acceptor end. So that particular amino acid gets activated. Okay, it gets activated in presence of ATP. What do I mean by activated? So to that particular amino acid, few phosphate bonds get added. So these phosphate bonds, when later on will be broken down, it will release energy, and this energy will be sufficient to form the peptide bonds between two adjacent amino acids. So that is how uh, you know energy is supplied, or the translation process is made energetically favorable. So this process of charging of tRNA is also called amino acylation. All right. Now, what is the site of the translation? It is ribosome. At the ribosome, the protein synthesis happens. So obviously, it will be the site of translation. And where is ribosome? Ribosome is in cytoplasm. So if you recall in the previous class, I told you that in the nucleus, replication happen, all right, as well as transcription happen. So the mRNA gets formed within the nucleus. And then mRNA, after it's formed, it gets transported outside of the nucleus and into the cytoplasm. And in the cytoplasm, there is ribosome waiting for it. Okay. As soon as a ribosome binds with the mRNA, the translation process happens or it starts. So now the ribosome consists of structural RNAs, okay, is also called rRNAs, and it also consists of several kind of proteins. To be exact or to be approximate, there are 80 different kind of proteins that are there in ribosomes. Now, in the inactive state, means when it is not synthesizing any protein, um, the two subunits, there are two subunits of the mRNA. One is small and one is large. Okay, there are actually apart from each other. All right. Whereas in active state, when the ribosome is going to start the protein synthesis process, it happens as soon as the smaller subunit gets in contact with the floating mRNA in the cytoplasm. It binds with the mRNA, then quickly the binding of the large subunit also happens, and it forms a structure like this. Here is the mRNA, is the smaller subunit, and is a bigger subunit. So both of these subunits actually hold the mRNA strand from the two sides. And now the uh, translation process can begin. Now the largest subunit has actually two sides, such that it can allow uh, you know two tRNA molecules to be present at a particular time. Why it is important? It is important because say at one particular time, at one particular time, at what the start of the translation process, one tRNA molecule with methionine attached to it comes. All right, and then uh, um, another site is vacant. Another site is vacant. So in that particular site, another tRNA molecule can now come. Say for that is carrying say another amino acid, say serine. Now these two amino acids needs to be in close contact with each other for formation of the peptide bonds. So that is why the two sides in the large subunit is helpful here. It allows this contact between these amino acids. When the peptide bond is formed, the first tRNA molecule that came initially, it leaves, all right, and the mRNA, it slides, or you can say the ribosome, it slides forward towards the 3' end. It slides. So it exposes another codon to the empty site. Now one site has become empty. And at that particular site, another tRNA molecule can come. So that is the translation, elongation step of the translation. I will show you again with the figure. Okay. Now, the ribosome has another function that it can also act as a catalyst. So basically, it will then increase the rate of the reaction for the formation of the peptide bond. Okay, it will, you know, it will increase the rate at which the proteins are formed. Now, in the mRNA, in the mRNA structure, we know at the 5' end there are caps, and the 3' end there is poly A tail. But other than that, there are also some kind of regions that are called a different kind of regions that you are hearing for the first time is called untranslated region, or UTRs. So there is a UTR at the 5' end and a UTR at the 3' end. And these UTRs, they are not translated, obviously, hence the name untranslated. Why UTRs are important? They are important to make the translation process efficient. Now, this is 5' end. This is the 5' end UTR. Downstream of this 5' end UTR is the sequence starting that is going to be translated. Okay. So AUG is there, is the first codon, the start codon from where the protein synthesis will begin. And it will continue until ribosome encounters the stop codon. Okay. Now the stop codon is there, protein synthesis stops. So 3' and UTR also doesn't get translated. So in this way, these two regions are not translated, but it's important for efficient translation. Now there are three phases of translation: initiation, elongation, and termination. So what are these phases, or what happens during these phases? This is an image, okay, that is showing what is happening during the initiation process. Now this is your small ribosomal unit. So when the small ribosomal unit encounters encounter encounters the mRNA strand, when it encounters the mRNA strand, it will start the translation process. Now this ribosomal unit, small ribosomal unit with it is also bringing the tRNA, initiated tRNA, that is having the anticodon and the amino acid methionine. Now this small ribosomal unit in this step, it has it has bound to the, you know, 5' end. It now then slides towards the start codon in this step. Now the anticodon in the tRNA has bound with the start codon in the mRNA, and thus now the initiation has begun, means now the translation has begun, because the large ribosomal subunit is now here, is now here to bind to the small ribosomal unit, and in between this is the mRNA molecule. So in this way, initiation complex is formed, but basically the ribosome is complete with both small and large subunits with mRNA in between. Now the sliding here, the mRNA will slide in this direction and ribosome will slide in this direction, as it is reading the codons in the mRNA and subsequently making peptide bonds and making the polypeptide chain. So that is the elongation step. Initiation step is that the formation of the initiation complex and the binding of the initiated tRNA molecule with the start codon. So these two means points actually signifies the initiation step. Next is the elongation step. Now this is the mRNA. This is the mRNA and it has codons. All right. So if this is the first codon, that is the start codon AUG, to which the initiator tRNA has bound, is bound. Now in this end there are two sites, right? So towards the 3' end, one site in the large subunit is now empty, to which another tRNA carrying another amino acid can come and bind. So in this step, the binding has happened. Now because of this binding, these two amino acid molecules are now in close contact with each other and form the peptide bond. So here transfer happens. This methionine forms a peptide bond with say another amino acid, say serine, and this is the small peptide chain that has now formed. And now this tRNA is empty. This tRNA is empty. So it has done its role of, you know, transferring the amino acid. So it will leave, and this machinery, a ribosomal machinery, will now slide in this direction, making the site A or this site empty again. So in this way, uh, ribosome is sliding towards 3' end, all right, whereas the mRNA is sliding towards 5' end. And as this sliding is happening, sites at the large ribosomal RNA becoming empty, or making space for subsequent incoming tRNA. And each of these tRNA is bringing the amino acid corresponding the codons that is being read in the mRNA. And these amino acids, they are placed in close contact with each other, that allows for peptide bond formation. Okay, and thus the polypeptide chain increases one amino acid at a time. It increases in length. So this is the elongation step. So when will termination happen? Termination will happen only when in the mRNA the ribosome encounters the stop codon. So for the stop codon there is no tRNA molecule. So here another uh, a particular molecule will bind, that is called release factor. The release factor here now binds to the stop codon, that now tells the machine to stop the translation process. All right. And because of this, the both units, large subunit and the small subunit of the ribosome, they detach from each other. Okay. The mRNA gets released as well as the polypeptide chain that is formed until now, that also gets released. So all the four things, two subunits of the ribosomes, mRNA, and the polypeptide chain, the newly formed polypeptide chain, all four things, they get released, and this is the end of the translation process. So that's it for today's lecture, where we have seen the importance of genetic code, right? And how genetic, because of genetic code, the translation step is possible, right? That is synthesis of protein from the mRNA. In the next class, we will look into regulation of gene expression, how gene expression gets regulated, as well as few other short topics, after which we will complete this chapter. Thank you for joining.